Homeostasis is the arrest of bleeding which involves a cascade of reactions. It is vital for the conservation of the body's blood supply. A typical blood vessel contains red blood cells, white blood cells, platelets, and proteins known as clotting factors, all produced in the liver except for factor VIII, which are all suspended in the plasma, the fluid component of the blood. The following reactions occur at the site of the injury: vascular spasm, platelet plug formation, and clot formation. The initial clot formed by the platelet plug is loose and unstable. Thus, clotting factors are activated to reinforce and make the clot more stable.
There are two cascading pathways involved in the clot formation: extrinsic and intrinsic pathways. The extrinsic pathway is first recruited and lasts for 8 to 15 seconds. The extrinsic pathway forms a weak or loose clot due to its short duration. Because of this, it recruits the intrinsic pathway. The intrinsic pathway lasts from 1 to 6 minutes. In the extrinsic pathway, tissue factor, with calcium acting as its cofactor, activates circulating factor 7 to factor 7A. Factor 7A can activate either factor 9 or the intrinsic pathway, or factor 10, wherein it now enters the common pathway. The extrinsic pathway forms a weak or loose clot due to its short duration, and because of this, it recruits the intrinsic pathway. The intrinsic pathway starts with the first plasma protein, called factor 12. It can become activated to factor 12A when it contacts with certain types of surfaces, including the collagen fibers underlying the damaged endothelium. Factor 12A then catalyzes the activation of pre-kallikrein and factor 11 into kallikrein and factor 11A. These proteins are anchored into the subendothelium by a high molecular weight kininogen. Kallikrein serves to amplify the activation of the intrinsic pathway by activating the neighboring molecules of factor 12. Once activated, factor 11A then activates factor 9 in the presence of calcium. Activated factor 9 then proceeds to form a complex with activated factor 8 and calcium, which binds to the phospholipid-rich surface of the platelet, where activation of factor 10 occurs. The activation of factor 10 is the point where the extrinsic and intrinsic pathway converge to form the common pathway of the coagulation cascade. Activated factor 10 forms a complex with its activated cofactor, factor 5A, in the presence of calcium, which converts prothrombin to thrombin. Factor 2A, or thrombin, then activates factor 1 to form an insoluble loose fibrin clot in the site of injury. Thrombin also activates circulating factor 13, which provides the necessary structure for a stable clot. Important to note at this point is the role of thrombin, as it further activates the factors 9, 8, and 5. Finally, fibrin is stabilized by factor 13, or the fibrin stabilizing factor. Fibrin is the biological glue that seals the hemostatic plug and ensures hemostasis. The formed clot is now mature. After formation, clot shrinkage pulls the edges of the damaged vessel together. This is known as clot retraction or syneresis. This process reduces the size of the damaged area. Over the course of 7 to 12 days, fibrinolysis will occur after the clot is mature. This dissolves the formed clot. Thrombin, the tissue plasminogen activator from the inner lining of the damaged vessel wall, activates plasminogen. Plasminogen produces plasmin, which dissolves the clot. The importance of factor VIII in clotting is emphasized by the fact that the disease hemophilia, in which excessive bleeding occurs, is usually due to a genetic absence of this factor. Clotting factor VIII deficiency is the most common form of the disorder. Like most recessive sex-linked X chromosome disorders, hemophilia is more likely to occur in males than in females. Female carriers can inherit the defective gene from either their mother or father. Hemophilia lowers blood plasma clotting factor levels of the coagulation factors needed for a normal clotting process. Thus, when a blood vessel is injured, a temporary scab is formed, but the missing coagulation factors prevent fibrin formation, which is necessary to maintain the blood clot. Individuals with hemophilia A or B are deficient in factor VIII and IX respectively. For this reason, patients with hemophilia cannot generate thrombin in sufficient amounts to induce normal clotting. Replacement therapy with factors VIII or IX can normalize coagulation. During hemorrhage, factor VIIa binds with tissue factor to form a complex. The complex converts factor X to factor Xa, leading to the production of small amounts of thrombin. However, due to deficiencies in factor VIII or factor IX in hemophilia patients, factor Xa is not generated, where it can be incorporated into the prothrombinase complex on the platelet surface. The deficiency in the platelet surface factor Xa may lead to the inadequate thrombin generation, preventing the generation of a stable fibrin clot. Hemostasis is essential for the conservation of the body's supply of blood. The constriction, platelet formation, activation of the extrinsic, intrinsic, and common pathways, maturation, and the fibrinolysis of the clot formation in the injured blood vessel are all important in order for proper hemostasis to occur.